Today we will be reading section 15.3, Applications of Genetic Engineering in Health and Medicine. In a way, biotechnology has always been a part of medicine. Early physicians used substances from plants and animals to cure their patients. In the 20th century, the use of vaccination saved countless lives. Today, recombinant DNA technology is the source of some of the most important and exciting advances in the prevention and treatment of disease. One interesting development in transgenic technology is golden rice. The rice contains increased amounts of provitamin A. A lack of provitamin A causes infant blindness and other problems. There is a hope that provitamin A-rich golden rice will help prevent these problems. Some scientists are making transgenic plants and animals that make human antibodies to fight disease. Several laboratories have made transgenic sheep and pigs that make important human proteins in their milk. Many of these proteins can be taken from the milk and used as medicines in people. Medical research. Transgenic animals are often used as test subjects in medical research. They can sometimes model human disease caused by defective genes. Scientists can study a disease and then use the models to test new drugs that may help treat the disease. Okay, the example up here, they are taking an engineered virus. They put in a gene to make normal hemoglobin. Disorders such as sickle cell anemia has a misshapen protein, hemoglobin, so it can't carry oxygen as well as it needs to. Well, this is a case of genetic engineering using a virus to carry that gene for the correct hemoglobin and put it into a bone marrow cell and then implant it into the bone marrow, and then the bone marrow start producing cells that have the correct type of hemoglobin protein. So that's one example of gene therapy being used to treat disease. So treating disease. Recombinant DNA can be used to make important proteins that can save human lives. Products now made in genetically engineered bacteria include insulin to treat diabetes, blood clotting factors for hemophiliacs, and human growth hormones. Scientists are now trying to learn how to fix faulty genes in people. Gene therapy is the process of changing a gene to treat a disease or a disorder. In gene therapy, a missing or faulty gene is replaced with a normal working gene. This process lets the body make proteins for enzymes it needs. How are genes added to human cells? First, scientists make a virus that cannot reproduce and hurt people. Then they put DNA that has the working gene into their modified virus. Next, they infect the patient's cells with the virus carrying the gene. With luck, the virus will insert the healthy gene into the cells that need it and correct the problem. Genetic testing. Suppose the couple wants to have children. Could they find out if they were carrying an allele for a genetic disorder? Genetic tests are now available for diagnosing hundreds of different disorders. For example, CF allele has slightly different DNA sequence than the normal allele. So CF would be cystic fibrosis. So genetic tests use a piece of DNA that is complementary to the defective allele to find that allele. Other genetic tests search for changes in cutting sites of restriction enzymes. So examining active genes. By studying which genes are active in cells, scientists can learn how cells work. Active genes make mRNA. So scientists use a test to see what kinds of mRNA are in different kinds of cells. DNA microarray technology lets scientists learn the activity levels of thousands of genes at once. A DNA microarray is a glass slide to which spots of sequenced single-stranded DNA have been tightly attached. Scientists then get mRNA from a cell that they want to study. They make and label single-stranded DNA from the mRNA. Then they add their labeled DNA to the wells of the array. They can tell which genes of the cell are active because the wells of those genes will also have labeled DNA. So key question. How can recombinant DNA technology improve human health? Today, recombinant DNA technology is the source of some of the most important and exciting advances in the prevention and treatment of disease. And for one example would be using the recombinant DNA to produce the corrected hemoglobin. They also use it to produce insulin and blood clotting factors. back there was the issue with getting human blood clotting factors to give to children with clotting disorders. A lot of times those human samples were contaminated, and then the children would get diseases from those contaminated samples. Well, if you're growing it in your own body due to recombinant DNA, then that also lowers the risk of contracting diseases from your treatments. So here's our vocabulary we can look at. Gene therapy is the process of changing a gene to treat a medical disease or disorder. An absent or faulty gene is replaced by a normal working gene. DNA microarray, a glass slide or silicone chip that carries thousands of different kinds of single-strand DNA fragments arranged on a grid. And again, they use those to see which genes are active. DNA fingerprinting is a tool used by biologists to determine whether two samples of genetic material are from the same person. And forensics is the scientific study of crime scene evidence. Here in vocabulary, we just mentioned DNA fingerprinting. This is a diagram that shows how it's done. One says chromosomes contain many regions with repeated DNA sequences that do not code for proteins. These vary from person to person. Here, one sample has 12 repeats between gene A and B, while the second sample has 9 repeats between the same genes. Restriction enzymes are used to cut the DNA in fragments containing genes and repeats. Note that the repeat fragments from these two samples are of different lengths. The restriction fragments are separated according to size using gel electrophoresis. Separated according to size. That's going to be important. The DNA fragment containing repeats that are labeled using the radioactive probes. This labeling produces a series of bands, the DNA fingerprints. So they apply electricity, and that causes almost like a magnet, causes charges to develop, and then it causes the genes, the DNA fragments to move. It's like trying to squeeze through something. Smaller things are going to be able to squeeze through more, and they're going to get further down the plate. So the smaller fragments are going to be further down. Larger fragments are not going to move as far. So then the arrangement of those fragments, how far they made it on that DNA fingerprint plate, through that, they're going through that gel. The bigger pieces have a hard time squeezing through, so they're going to stay closer to the top. They are not going to go as far. And then the plate can tell you how many big fragments and how many small fragments, and they can compare. Remember, enzymes are specific to what they're going to cut, so that's what the restriction enzymes, by using particular restriction enzymes, they know right where those enzymes are cutting, and then they can compare those and see if the DNA matches. So personal identification. The human genome is so complex that no individual has DNA exactly like another individual. Identical twins are the only exception. Molecular biology has used this fact to make a powerful tool called DNA fingerprinting that is used to identify individuals' DNA fingerprinting DNA fingerprint analyzes pieces of DNA that may have little or no function in a cell. However, these pieces, called restriction fragments, vary widely from one person to another. How do scientists make a DNA fingerprint? DNA samples can be taken from blood or any other body tissue. The restriction enzymes cut the DNA into pieces. Next, gel electrophoresis separates the pieces by size. A DNA probe finds the fragments that have highly variable regions. This is shown in a series of DNA bands on the gel. If another enzyme or probe were used, the patterns of bands from one person is unlike anyone else's patterns or bands. So forensic science. DNA fingerprinting has been used in the United States since the late 1980s. It has been of great help in forensics, the scientific study of crime scene evidence. DNA fingerprinting has helped solve crimes, convict criminals, and even overturn guilty verdicts. To date, DNA evidence has saved more than 110 persons who were sentenced to death for crimes that they did not commit. DNA forensics is used in wildlife conservation as well. African elephants are highly vulnerable species. Poachers kill the animals illegally for their valuable tusks. To stop the ivory trade, African officials now use DNA fingerprinting to identify the herds from which ivory has been taken. And we'll look at some examples of DNA fingerprints and some handouts we're going to do in the next few days. And what we'll look at, just have a few bands. It's going to be rather simple. But as you can see right here in this picture, they're really much more complex than that. Okay, so there's hundreds of bands there to look at. So it's not simply the 3 or 4 bands that we'll be looking at, but that simplifies it for us to be able to learn. Okay, so establishing relationships. How can we figure out who is the biological father of a child? DNA fingerprints makes it easy to find alleles carried by the child that do not match those of the mother. Any such alleles must come from the child's biological father. The alleles will show up in the father's DNA fingerprints. When genes are passed from a parent to a child, the genetic recombination mixes up the molecular markers used for the DNA fingerprinting, so ancestry can be difficult to trace. There are 2 ways to solve this problem. The Y chromosome never undergoes crossing over, and only males carry it. So Y chromosomes pass directly from the father to son with few changes. The small DNA molecules found in the mitochondria pass from the mother to the child with few changes. Mitochondria are the organelles that release energy from sugar to power cells. They also carry DNA. It rarely changes. The DNA in the mitochondria is passed from mother to child in the cytoplasm of the egg cell. These genetic markers can be used to trace ancestry for many generations. Because mitochondrial DNA is passed directly from the mother to the child, your mitochondrial DNA is the same as your mother's mitochondrial DNA, which is the same as her mother's mitochondrial DNA. This means that if 2 people have an exact match in their mitochondrial DNA, then there's a very good chance that they share a common maternal ancestor. Y chromosome analysis has been used in the same way and has helped researchers settle long-standing historical questions. Key question: how is DNA used to identify individuals? DNA fingerprints analyze sections of DNA that may have little or no function, but vary widely from one individual to another. Okay, so checking understanding. You're going to answer these questions. So you're going to pause, try to answer them on your own, and then we'll talk about them. Applying vocabulary. Use the highlighted words in the lesson to complete each sentence correctly. An absent or faulty gene is replaced by a normal working gene using, and that would be gene therapy. Number 2, the study of blood or other crime scene evidence is called forensics. Forensics is studying the crime scene evidence. What is golden rice and how has it helped people? Okay, golden rice is rice that has been genetically engineered to produce provitamin A. And it's helping by, especially in poorer countries, third world countries, people don't have a lot of nutritious food. They eat a lot of rice, so if you give them the provitamin A rice, it helps prevent blindness in those children if they can get enough vitamin A. Blindness and other illnesses. So what are the steps of DNA fingerprinting? You have to splice the DNA, charge it, place it on the plate, apply an electrical current, and then analyze how far the fragments move. And you can see who has bigger fragments and smaller fragments that are unique to the individual. Okay, number 5, it says write a paragraph that answers the chapter's mystery clue. So here's our mystery clue, and you can write a paragraph. Open your doc and write a paragraph. Open a doc and answer these questions on that doc. Write a paragraph. Mystery clue. What kind of evidence do you think investigators collected at the crime scene? What kinds of tests would they have to run on this evidence? What would the tests have to show before the suspect was released? So be sure to answer and turn in those questions. You can make a doc and turn those in.